Okay, so let's, we're going to start on the microstructure with neuron anatomy. We're going to draw a neuron, which we have done before, but we're going to add a little more detail to it this time. Okay, so I'm going to start with just these weird little branches here. Okay. So what we have here, this is a neuron. This is exactly what it looks like. Okay. And the way that the signal passes through the neuron is it's going to start here to the left and go to the right. Okay, which is important. Orientation helps us determine the anatomy on this in this instance. And what we have here are a lot of these processes on this left end here, and these are called dendrites. All right, and so this is the receiving signal. What you would have here at a dendrite is you would have the connection of another neuron upstream of that. And if it was passing the signal, then it would go through that neuron, and then it would start at this dendrite. Okay, but it could occur on any one of those dendrites. It doesn't have to be that one that I highlighted. All right, and then the dendrites lead into the larger section, which is the cell body. Okay, and in the cell body, you're going to have a nucleus, okay? And the nucleus is going to manage this signal and kind of figure out whether it's strong enough or frequent enough to pass it or to ignore it. All right, there is a section here, and I didn't draw it very well, so I'm going to kind of redraw this. So really the transition here between the cell body and this is called the axon. There's this kind of triangular portion here. This is called the axon hillock. All right, and then, of course, the whole cell is surrounded by a cell membrane. Okay, do you remember what the plasma membrane or cell membrane was called in the muscle cell? Sarcolemma, okay? And so in the neuron, it's called the Axial Lemma. Yeah, so there's some consistency. We didn't make up a brand new word. We just stuck a couple things together, right? Makes sense, good.
Hold on, the axial lemma is what?
That's the plasma membrane. All right, but it's specific to an axon. So an axon cannot have a sarcolemma, right? That would be a muscle cell. All right, at the end of the axon, we then have, use red again. Our terminal branches. All right, and what did the terminal branches meet on our muscle cell that we talked about last time? Okay, yeah, it would go to a different synaptic cleft. So they would go and meet different muscle cells, right? But this isn't, we aren't going to deal, you could have a neuron going to a muscle cell, which we showed last time. But it could also just meet another neuron, which is what we showed at the beginning up here. But at the very end then, what was that structure called? Yes, the axon terminal. Right, so there's all of these axon terminals at the end, very end of our axon. All right? And this is where then another signal would occur or be passed. And so sometimes we would call this area the synapse.
So you call either the axon terminal or the synapse?
Yeah, let's do a slash.
So there's multiple synapses.
Yes, synapses would be plural, right?
Synapse.
Good. Yeah, so we've got 4 axon terminals, 4 synapses at the end of our axon here. Yes?
So the axon terminals are what go down into like the whole diagram we did last week, or last unit?
Uh-huh. Yeah, so the axon terminal was the very end, which started our process of initiation.
So that
Right?
little like part that came down at the top is the same as the axon terminal. It is the axon.
Exactly, right. And so we have a synapse right here.
Oh, yes.
Nice, Jack.
Nice question,
Right. This would be the synaptic cleft. Yeah, it's all coming together. Good. Okay. Now there are a few so this is all the stuff on the axon. There are a few accessory structures which are important to some neurons, not all, but some. And we're going to draw just this little sheath, okay? And it's going to go all the way down our axon. And it's made of a fatty substance called, I heard it, myelin. So it's the myelin sheath. Jack, do you want to teach today?
I'll take the next class.
Okay. All right, and the myelin sheath is made by one of 2 different types of cells, depending on whether it's the central nervous system or the peripheral nervous system. Okay, we've got the Schwann cells. Or the Schwann cells is going to be peripheral nervous system. And then the oligodendrocytes. Yeah. Is central nervous system. Okay, we didn't give ourselves much room here. But essentially you have this cell, and he's got little processes. And it made the myelin sheath as it wrapped around those different ones. So we'll say this one's an oligodendrocyte. Yes?
You've drawn it where one oligodendrocyte covers, like makes multiple sections of this sheath. Will they also cover more than just the one axon? Will they get other axons?
They could, yeah. If there's
Here, but
right, if there's lots of little axons in one area, it doesn't really differentiate one cell to the next. It just grabs on, yeah. Okay, and that's our neuron. Okay.
can you zoom up? Just
Well, which one? Back here?
up and get it all in there. Yeah. Perfect.
Good. Okay, and you guys already started to fill in some of the functions of these, but let's see how much you remember. So I'm going to pause this. So surrounding your brain, you don't have blood. Okay, you don't have blood. Instead, you have cerebrospinal fluid. All right, and if we were to characterize cerebrospinal fluid as a tissue, what type of tissue would it be? Before you answer that. Okay, hold on. All right, so what it does is it serves a similar function as blood. It is a place for delivering nutrients and taking out metabolic waste and surrounding the cells with what they need. So very similar to blood. All right, so what tissue would it be then? Yeah, it's a fluid connective tissue. We probably should have added that to our list way back when we were talking about tissues. All right, so we'll discuss why we have this in a minute, but a structure which is very important for this is called the choroid plexus. Okay, and that is, you can see it as this red part here, just underneath our Nike sign. What was that called? It's not labeled, so you guys should remember though, right? Yeah, the corpus callosum, okay. There's another choroid plexus down here.
Oh, hello.
Yeah, a little red guy down there, okay. And what's happening here, these are what is making our cerebrospinal fluid. And they're made by a glial cell called the Ependymal cells. So the ependymal cells help form the blood-brain barrier and produce cerebrospinal fluid. So you'll find these abbreviations a lot in the text and in literature. So those are good ones to know. Blood-brain barrier and cerebrospinal fluid. And you don't have to know everything in this figure. I just wanted to illustrate how the cerebrospinal fluid, as you can see, the arrows are showing the flow of the cerebrospinal fluid. So it comes out of the choroid plexus underneath the corpus callosum into what's called the lateral ventricle. It's a big space filled with cerebrospinal fluid. It then flows through this cerebral aqueduct into the fourth ventricle. You have more cerebrospinal fluid being made at this lower choroid plexus, and then it goes all the way around the brain and is constantly bathing the brain in cerebrospinal fluid. Okay? Now, there's another glial cell which is important for this mediation between blood and brain. Okay, and the second one is called an astrocyte. Okay, and its actual contribution to the blood-brain barrier is actually less than what they thought of 5 or 10 years ago. But it still mediates, we'll just call it the mediator, functions of neurons. And by mediates, it has glycogen stores, so it can release glucose if the neuron needs energy. It can pull waste products away. It can add neurotransmitters to neurons, okay? So it does a number of different things helping in regulating the function of the neuron. And they are abundantly found throughout nervous tissue. Yes?
So, the choroid plexus is where the
Yes.
cells are found?
Yes. Ependymal cells are concentrated at the choroid plexus. The astrocytes are found everywhere.
Sorry,
Yes.
the way it's drawn, it looks like there really isn't blood around the brain.
Right.
Is that true?
Yeah, so there is blood that goes, blood vessels that go around the brain, but they don't directly contact. So they are found within the meningeal layers, okay? But this is, that is superficial to this,
So you
these
get
structures.
hematoma in that layer. You don't get hematoma in your brain.
Right, yeah.
But there's blood within the brain?
There's no blood within the brain. There's only cerebrospinal fluid.
So then,
It's around the brain. And it exchanges with, it still exchanges with the cerebrospinal fluid
at the choroid plexus.
at the choroid plexus.
That's where your artery, your big time blood supply goes to the choroid plexus, and
Right,
then that takes the nutrients from your blood, puts it in the cerebrospinal.
right. And why would you want this to yeah, well, I'll answer your question
Well,
first.
I asked a question. So if there's not actually any blood in the brain, what is the significance of the damage of, like the stroke is explained as damage to the blood supply?
Right, the blood is still vital to the function of the brain, okay? But it has to first exchange with our cells, our blood-brain barrier, to get those nutrients in, and then it comes in as cerebrospinal fluid. So it's just like another step. And so there are a lot of things that are in blood which are not in cerebrospinal fluid. Okay, so it's a more limited supply of
This might be
things.
some question, but is cerebrospinal fluid also red? Like-
No, it's actually, no, it's more clear, right? So, yeah.
I just asked because like someone gets hit in the head, it bleed, it looks like. But that
There's
might-
still plenty of blood around your brain.
Okay,
But yeah, inside your brain, no.
How does the nutrients you get from your blood to the
Through the ependymal cells, through the choroid plexus.
So it's like a filtration.
Exactly, it's filtration system, yes.
Okay, so I'm still a little bit confused with the ependymal versus the astrocytes. 'Cause
Okay.
in the video you said that the astrocytes made the brain blood barrier.
Right, which is what your book says, and which is fine. But when I was looking more into it, it actually appears to be a little bit less of what it does and does more of a lot of other things. Yeah.
So we should.
Good question.
Paying attention to the notes from the video lecture, we should pay attention to this.
You can
I'm not saying like the entirety, I'm just saying like this.
No, no. No, you can say it's part of the blood-brain barrier.
Okay.
You can't say that. It does help with that. And if that's all you want to know, that's fine. I'm just telling you more accurately, it does a lot of other things as well.
Okay.
But good questions. Any other questions? Okay. So my next question is, an astrocyte in the peripheral nervous system are satellite cells. Okay, which also surround the neural tissue and mediate the exchanges between the blood.
Ependymal cells only in the CNS.
Right, ependymals only in the CNS. So we can then from here go back to our worksheet, right? Okay, so here is an article from the primary literature. It's called the blood-brain barrier. So if you want to learn a lot more about the blood-brain barrier, you can read this article. But what I want you to do, you and your neighbor, is just read this first paragraph of the abstract. And I want you to answer this question. Why is there a blood-brain barrier? Okay, so I'll give you a few minutes to do that. Do we need to read it again? Okay, exactly. So we value our brain tissue more than we do the rest of our body. I know that's I can't even say that word. All right, so we protect our neural tissue more than we do the other parts of our body. All right, so we are going to filter and create a second system to keep out, like it says, protects the neural tissue from toxins, pathogens, alterations of barrier properties that are an important component of pathology and progression of different neurological diseases. Okay? So if you have an infection and those bacteria are in your blood, we have a second line of defense to keep them from getting in your brain. Because if your brain is infected, you're dead. Okay? Right. So, and there are also a lot of other toxins in your blood that are being distributed and filtered out, through your liver or through your kidneys. And we don't want those to affect how your neurons are synapsing, how they're functioning as well. So we have ependymal cells, we have astrocytes, the equivalent of, and we've drawn out a specific type of neuron. What we actually drew out is called a multipolar neuron. Where it had lots of dendrites connected to the cell body, connected to an axon. All right, multipolar neurons are the most abundant. Their function is an interneuron. Okay, so they are in between sensory and motor neurons. And the site of the sensation or the site of the motor function, okay? Or between the brain, right? So they're going to be where the signal is being propagated. Right?
You can find a lot of these in like gray matter because they're just
Right, they're right there to pick up the signal, send it. Yeah.
It's not; these aren't the long transductors.
These are long ones as well.
Oh, okay.
Yeah. Right? So then we have two other types which are less abundant. We have a unipolar neuron. Where you have a cell body, and then right off the cell body it branches, going in both directions. I should give myself more space here. But you still have, dendrites on one side, and then the axon terminal on the other. And so the signal would go like in this direction. All right, these are associated with sensory TPPT. Okay, that stands for touch, pain, pressure, and temperature. All right, so where would you find these? Where would you find a lot of these? Skin. All right, good. Yeah.
Where's the axon hillock for that?
Yeah, so it still has like this axon hillock there.
So it has to like go down into the cell body and then back out?
Yeah, I think the cell body is less important to the transmission as it is in the multipolar neuron. So it's still,
To get clearance from the axon hillock to move on.
yeah, not in this instance. So they function differently. Most of them function like we talked about before.
That makes sense if that's the most common.
Yeah. All right, then the last one is bipolar. And so it has dendrites on one side. The cell body in the middle. And the axon on the other side. These are also sensory neurons. Okay, and my acronym for this is VHS. Anyone know what a VHS is here? No, you guys have no idea. You're too young. No, you don't. Just kidding. All right, so vision, smell, what is H? Hearing, thank you. Whew. And taste, right. So on a VHS, you can experience both vision and hearing. Not smell or taste. Yeah. Yeah, I guess, yeah. Okay. We'll go more into bipolar neurons when we talk about the function of, or how the optic nerve gets signals from the retina. So you find those, of course, in the retina. They're also associated with our other nerves.
So would you say
Yeah.
that the bipolar neuron is more common in the unipolar neuron, but not as common as the multipolar neuron?
The multipolar neuron is definitely most common. I'm not sure those two in relation to each other.
You have more skin than you do eyes.
You do, yeah. Yeah, these are much more specialized, so that would make more sense, yes. So you would, if you were to rank these, right, these guys would be number 1 in abundance. These guys would be number 2, and these guys would be number 3. Gold, silver, bronze. But you don't have to know that. Let's just answer your question.
You never know.
You never know. Okay. Until you don't. Okay, so those are different microanatomy of different neurons there. Okay, last thing we're going to talk about, myelination, right? So how myelin occurs, and what are the two types of cells that myelinate nerves? They are glial cells, oligodendrocytes, and Schwann cells. Okay, good. All right, and it's just 3 simple steps to myelinate. So step 1, which is also right here, right? The Schwann cell, we'll say Schwann since that's easier to spell. All right, it attaches to the neuron. All right, once it attaches, then it wraps around it multiple times. Okay, you can see that it like circles around and around. Okay. And then the third part, it forces the cytoplasm out. So what you're left with is many, many layers of what structure? The plasma membrane, more specifically known as the axial lemma, good, which is made of lipids, right? This is the phospholipids, okay? And that lipid structure allows it to repel the ions, which is what the signal is made out of, okay? So it allows that signal. So you can see on this, it looks like an electron microscope image, all these layers here. Okay, so this is a cross section. Yeah.
The plasma membrane of the neuroglia is also called the axial lemma.
Oh, yeah, that's a good point.
So, because you're not.
Because they aren't technically neurons,
Right, because you're not.
but it is a plasma membrane. Yeah.
Right. I was just confused about which cytoplasm was being squished out.
It's cytoplasm.
It's just the cytoplasm of the neuroglia. It has nothing. It doesn't do anything to the inside of the axon.
Correct. Yeah, the good clarification. So it forces the cytoplasm out of the Schwann cell. So it reduces the amount of cytoplasm between those different layers. So now you just have stacks of layers on top of each other.
Whether it's axial lemma or not.
Right. So that's not technically axial lemma because they aren't technically neurons. They're just helper cells. Okay. Any other questions? Yes. So the question was, how are your glial cells made? They have different derivations. So for example, the microglia come from the immune system, but they aren't from the nervous system. So I'm not sure exactly the other ones, but I do know that one comes from the immune system. Yeah. Any other questions? Okay, you got 5 minutes extra to study on your own. We're done.